Everybody, and welcome to Chapter 9. We're going to be talking about microbial nutrition and growth in this chapter. Where do our microbes get their nutrients from? Where do they get their energy from, and how do they generate those things? So these are our learning objectives for the chapter, so keep them in mind as we go through this content. Be sure that you know enough about each of these learning objectives that you would be able to have discussions about them. Right, starting off with how our microbes get their energy. Some microbes get their energy from sunlight, and some microbes might get them from chemicals. In case we were talking about all living organisms, this is information that applies technically to all living organisms. And there are additional categories that we could apply microbes to, but we're going to stay in these basic ones here. So organisms that get their energy from sunlight, we call phototrophs. Troph is referring to food. And so where are we getting the energy from our food source? In this case, if we're phototrophs, we're getting it from the light. Sunlight, quite literally, is being converted into usable energy for the organism. And so this is where we'll start learning about the importance of photosynthesis in generating the energy for the organism. In Chapter 10, we're going to break that process down more in more detail. Then we have the chemotrophs. These are organisms that break down chemicals, and by doing that, are able to release that energy and then use that energy for the cell's functions. So this is where they're getting their energy from, not their carbon. So that's going to be different terminology for that. So we talk about carbon, that all organisms that are alive that we're aware of need carbon, but what other elements are essential? And so this would be chomps. So there are definitely more elements that are present in living organisms, but these are the major six. This is carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And remember, phosphorus and oxygen come together to create phosphate. Those are different things. Phosphorus is the P in the phosphate, and phosphate, PO4, is going to be a molecule, and that is made up of those elements of phosphorus and oxygen. Then we can break things down with our nutrients as the macronutrients, things that we need in larger quantities, things that we would consider things like protein and fats and carbohydrates in our diet would be our macronutrients, but we also have micronutrients. We still need these guys in the micronutrient area, but we need them in smaller amounts than the macros. So this would include even things like copper or iron in our diet. We need them in smaller numbers, but they're essential for enzyme function, and you can die if you don't get them. We need them in such small amounts, but we still absolutely need them. And this is true for microbes as well. They might need different ones than you or I need, but they are still essential. So then this gets into the carbon source. So starting off with chomps, right, keeping that CHONPS in mind, where are we getting our carbon from? And so we can still refer to our organisms as troph, but just now keeping in perspective carbon sources. So remember, we first talked about energy sources, light or breaking down chemicals as sources of energy, but now we're talking about carbon sources. Organisms that break down existing organic molecules to build their own organic molecules, like us. We break down like the protein or the fats or even the DNA and stuff from the food that we eat in order to get our carbon sources. And so those are the heterotrophs. They are using existing organic molecules that were made by other life forms in order to provide themselves carbon. Whereas the autotrophs, which I already started writing something on this one, the autotrophs are referring is basically to where they're getting their carbon, they're making the carbon from inorganic sources. So when we talk about inorganic, it's important that we remember with inorganic, if you do not have hydrogen bound to that carbon, it's inorganic. So it's not about whether you have carbon, it's whether you have a hydrogen bound to your carbon. So carbon dioxide is inorganic. Plants take carbon dioxide and convert it into glucose. And so their ability to do that means they can turn inorganic carbon dioxide into an organic carbon compound like glucose. And so that's a pretty unique thing to things that can go through photosynthesis, using energy from the sun to turn inorganic carbon-based carbon dioxide into organic carbon, things like glucose. All right, so this again is breaking down the difference between where we're getting our energy from phototrophs. So again, these are all trophs. Okay. So phototrophs are getting energy from the sun. Um, and then we have the um, chemotrophs that are getting energy from chemicals, obviously. Breaking down a chemical gives you energy. And then where we're getting our carbon, we have the heterotrophs and the autotrophs. Hetero, we're using existing organic molecules. Autotrophs are using inorganic carbon to make their organic molecules. So those are our breakdowns, and we could combine these words. We have a chemoheterotroph. That's us. Energy from chemo, chemicals, carbon from hetero, organic, existing organic molecules. So that's us. We are chemoheterotrophs. Photoautotrophs, energy from the sun, and then carbon from inorganic molecules. So it's two different aspects. Where's their energy coming from, and where's their carbon coming from? So keep that in mind as we learn about our organisms. All right, growth factors. Coming back to this again, we learned about this previously when we were talking in chapter 3 a little bit about learning about different types of media, that a growth factor is a compound that needs to be provided to the organism that the organism cannot make for itself. And so it has to be provided by their environment. Okay, in this case, we're talking about their media, right? And so that was what we learned about chapter 3. In in their environmental, where they're getting nutrition from in the wild, it has to be provided by their diet in their environment. So it's not always media, right? It just depends on how it's being used. Remember, growth factor is essential to our fastidious organisms. Any media that we add the growth factor to would be enriched. All right, so photoautotrophs, they get energy from the sunlight. They produce organic molecules that can be used by heterotrophs by using inorganic carbon. Okay, so they produce that for themselves. And we call them primary producers. You may remember this from like middle school biology or something like that. The primary producers basically produce the organic compounds through that autotroph process, which is powered by the sunlight here. Then we have the chemoheterotrophs. This is basically going to be probably a majority of our microorganisms, and probably we get used to any animal is a chemoheterotroph, basically able to break down chemicals for energy, and then they're using organic compounds made from our producers in order to build our own molecules out of those components. Okay. So there's another category that we call saprobic organisms. This is kind of like a subtype of the heterotrophs, but they're not feeding off of like living targets or anything like that. They are specifically breaking down or decomposing dead matter. And so this could be dead plant matter, animal matter, dead animals, microbes, anything that's non-living and that we are decomposing. Our saprobes are what break those things down. And so they usually release enzymes into their environment, basically. And when they release those enzymes, that basically exuding those enzymes into the environment, that those enzymes will then break things down. So if we have organic debris, dead plant matter, leaves that are in your backyard or whatever it is, that they can secrete enzymes, which is here in the like red in this image here, that basically break that down. And now they can absorb those nutrients through their cell membrane and whatever. So that's how saprobes will break down what's outside of them and then absorb those nutrients, which leads to the decomposition of the basic overall structure of whatever that dead, organism is. Okay, and so all organisms have to have a way of getting their nutrients into their cells somehow. And this can rely on basic diffusion, or it can rely on specialized mechanisms for transporting things across the cell membrane. This is basically relying on this idea that the cell membrane and the cell membrane is very selective. The cell wall is a little selective, but very non-specifically. So it's pretty Non-specific. So we really rely on what can cross the cell membrane because the cell membrane is selectively permeable, which you may have heard this term before. The cell membrane is selectively permeable. So we'll come to that in a little bit because that's important. The first principle we have to understand before we get into how things cross the membrane is just general diffusion. Diffusion is the movement of molecules basically with a gradient. So in a gradient fashion, so from an area of high concentration to low, and we're trying to get it all equal everywhere. So that's what physics has us wanting to do, is that molecules will be equal everywhere. And so that tendency for that to happen, we call that entropy. And so this basic law, it's one of the laws of thermodynamics. So the idea is that molecules want to move from that high area where they're all clustered up and built up to an area where there's none. Think of a big pile of bouncy balls. They're going to roll out and spread out everywhere. That even happens on that larger scale. So that tendency to want to spread out and not basically just collect in one spot. Idea of spreading out is diffusion. It's just like if I spray perfume at one part of the room, eventually even the back of the room will smell it as those molecules diffuse through. So that's the idea of diffusion. But if the molecules can't move through a barrier, then that can control how the molecules will move. And in fact, your membrane, your cell membrane, and the cell membrane of our microbes basically can control what can and can't move across it. The membrane itself is selectively permeable, or another word for that is differentially permeable. The only molecules that can move across the cell membrane is water and certain very tiny molecules. But most importantly, water. Water can diffuse across the membrane. Proteins can't. Lipids can't. Nucleic acids can't. And that's what keeps the stuff inside the cell, but it's also what keeps outside the cell out. So that controls what's inside versus what's outside the cell is basically just the membrane existing. But water can cross. So this is really important because now whenever we put cells into solutions that may have, stuff dissolved in them, solutes dissolved in water, which is the solvent of life, that whenever we look at that system, basically can determine that if only water can move, we can't move the other molecules, can basically control what's happening inside of the cell. The cell will have to accommodate that change. So the movement of water across a selectively permeable membrane is osmosis. So most importantly, the diffusion of water is osmosis. And in living systems, what controls the diffusion of water is the fact that the membrane is selectively permeable. So that's the concept of osmosis, the diffusion of water. And in living systems, the diffusion of water depends on a selectively permeable membrane. You've probably seen images like this, but it's literally only governed by the fact that only the water can move, not the solutes. So remember, life, the solvent of life is water. The thing that is dissolving the liquid is the water. The thing that's dissolved in the liquid, like salts and things like that, are solutes. So you need to remember that term, all right? And they basically, if the solute, like salt or something like that, can't cross the membrane and only water can. And what the chemistry of biology wants is for everything to be equal on both sides. It wants the same concentration of salt inside the cell as outside the cell. And it's not just salt. That applies to proteins, it applies to sugars, and all the time. It wants everything to be the same on both sides of the membrane. And so water will move across the membrane to try to always keep that balance there. So those are the tonicity aspects of how that stuff works. And this is an image that kind of shows that if we have some things collected inside of this balloon that we put into pure water, the red molecules are our solute and the blue molecules are the water. So if you have salt water balloon that you put in the water that's made up of your cell membrane, and you put that salty balloon into the water, and basically what's going to happen is the water's trying to water down what's in the balloon to make it equal outside. Can it? No. So that cell is going to keep taking in water to try to accommodate that, and eventually could burst the cell open as it fills up. The opposite situation can happen where you take a mostly water cell, pack it into salt, and what happens? It draws the water out of that membrane. The water moves, the cell does, or the salt doesn't. The water moves, the salt doesn't. And so that can govern what's happening in the cell. And this is extremely important whenever we're administering fluids to our patients. We have to think about the effect of those fluids and in what volume we're giving them in relation to how that stuff is managed. Some of that stuff is managed on that cellular level. Other stuff is managed on the macro, on the organism level in your body by your kidneys. So trying to keep your blood the same concentration of stuff as what is in your blood cells so that your blood cells stay happy and don't burst or shrink too much. That's your kidneys' job. And so all of that would be managed by your kidneys, basically. So isotonic conditions are whenever things are the same inside the cell as outside. Is water moving across the membrane? Yes, but water is moving in and out at the same rate. It's just pretty much balanced. We move a little bit out and a little bit in. Water never still. It's never still. They're not doing nothing, even when it's equal concentrations on both sides. Then we have hypotonic conditions. And hypo means low. And this is not talking about the cell typically. I'm talking about the solution outside of the cell. So when we have a hypotonic condition, this is referring to it being hypo, low salt, or low sugar, or low solute outside of the cell, causing water to move into the cell to try to water down what's inside of the cell, causing the cell to swell up to accommodate it, and that can lead to bursting open of the cell. So that net direction of osmosis is water into the cell in a hypotonic solution. So increasing the volume of the water in the cell to try to water down the cell to match what's outside of it. Okay. This is like putting the cell in pure water. That's the most extreme example of a hypotonic condition. Hypertonic is hyper is more, is higher, right? And so in this case, we're putting osmotic pressure on the cell. And how that's done is higher solute outside of the cell than is inside it. And again, now the cell is the watery one, and the outside is lower water. So what happens with the water? Moves out of the cell to try to balance it out. If it's too much solute outside, then that can shrink the cell so much that it could be detrimental to the cell. But we call what the effect of a hypertonic solution on a cell, like packing a cell in salt, for example, osmotic pressure. That's the effect of drawing the water out of the cell, osmotic pressure. And this can be used to control microbial growth. More effective because some microbes have adapted to live in fresh water, to get rid of the water they keep taking in. They'll have mechanisms to get rid of it. But many microbes can't tolerate the high salt. Microbes that have sturdier walls are more likely to be able to maintain their shape and not shrink up too much. Gram positives, maybe. Just saying. So keep that in mind as we learn about our microbes moving forward. So here we can see that we have examples of these different conditions in an isotonic solution. It's equal on both sides. The water is just diffusing equally. In a hypotonic, remember, this is like a pure water situation. You've got stuff in your cells. It's not pure water in your cells. But outside of your cells is hypo because there's nothing except water outside. So what happens? The water outside tries to water down what's inside of your cell. The cell takes up water and it can continue to swell until it bursts. Okay. In hypertonic, we have more solute outside of the cell, so high salt outside, more water inside. So what happens is only the water can move. So the water moves out of the cell to try to water down its outside environment to make it equal. If that happens too much, the cell shrinks too much. This can cause something called plasmolysis, the excessive shrinking of the contents of that cell. And that can be detrimental to the survival of the cell as well. Not just bursting with too much water, but shrinking from not enough water can be detrimental to cells as well. We don't just rely on osmosis for things to move across the membrane. Relying on just osmosis and the ability of molecules to move across the membrane naturally is just simple diffusion. But we can also allow molecules to move from high to low concentration. If they can't cross the membrane, we can have special channels built in the membrane that would allow diffusion to happen. So this exists that we can actually build certain channels in the membrane to allow for passage of molecules by diffusion across the membrane, and we call that facilitated diffusion. This can even involve binding to a receptor and then moving by diffusion. That would allow the channel to be controlled. Or it could be open channels. It just depends on what we're moving and why and how. So typically it's controlled in some way, thereby allowing the cell to kind of control what's in and out of it. In fact, one of the mechanisms that some of our immune system uses to fight off bad guys is by poking holes into microbes. Our immune system can build holes in microbes, and if they can't control what's in and out of their cell, then we will facilitate the diffusion of what's in their cell into the environment and vice versa, which is deadly to those cells pretty quickly. And so that's a forced facilitated diffusion on our end. In this case, what we're talking about here, you can control diffusion, still moving from high to low concentration, by having channels that are basically like triggered to open or close based on the presence of molecules that they interact with. And this doesn't rely necessarily on energy. It just relies on there being a channel that's triggered by diffusion. Here we have active transport. So in this case, we would need energy in order to move the target molecule across the membrane, because this is usually either going against the gradient or wanting to go faster than diffusion allows. So for example, if you're trying to eat up nutrients, you might already have more nutrients in your cell than what is outside the cell, but you want more of it. So you would be to add energy in order to grab it rather than let diffusion work, because diffusion wouldn't work because you're going against a gradient, right? So so there's that. Or if you just want the nutrients faster or interact even faster, again, would require energy. Anytime that you aren't allowing diffusion to just work naturally and you want to help it along, you're going to need energy to power that. And so that's active transport. And in fact, many of our nerves and how they fire rely on very controlled, interaction with electrolytes like sodium and potassium, and even hydrogen ions that we're talking about in our electron transport chain. Now, all of those will rely on special pumps and channels and things like that on the membranes that kind of force things against a gradient and allow our nerves or our aerobic respiration to take place. Okay, and so again, carrier-mediated active transport, where we have are trying to take up more molecules, is going to require energy, and that's typically going to be in the form of ATP. ATP is the number one energy source in the cell, and we'll talk about how ATP is generated in chapter 10, so it's coming up soon. So active transport, there's other examples of active transport. If we're doing bulk, like if we are an amoeba and we're just going to circle our little amoeba fake feet around something and then eat it up, then that's a bulk transport process. It's going to need even more energy in order to make that happen. So endocytosis is the process that we use to do that. That could be in large ways, like with phagocytosis, or small ways, like we see with the cilia and the microvilli and things like that, like in our intestines. We take small amounts of little sips of things. It's called penocytosis. But both of those are a type of endocytosis. Endo means into, cyto means cell. So getting things into the cell in kind of a bulk way, not by molecule, but in kind of bulk. All right, things that can influence our microbes, they can be dependent on temperature, gases in their environment, and pH. Their osmotic pressure, how much salt can they tolerate in order to live? Radiation, pressure of their environments, which we call hydrostatic pressures, like the barometer, right? That sort of stuff. Moisture presence, and then presence of other organisms can all control how organisms live. So we're going to be talking about that next. So we have the temperatures, which is the cardinal temperatures, basically are referring to the preferred temperature at which microbes will grow. So we have a minimum to a maximum for our optimum temperature growth. So those are our cardinal temperatures. And this basically can be used also as a characteristic for identifying our microbes. It's basically their favored temperatures for growth, okay? So we have all sorts of different groups of microbes that we can classify based on their preference for temperatures. And so in this chart, we can see different groups that are basically defined by these different colors of lines. This blue dotted line here are organisms that prefer temperatures that are below 10 degrees. These are in Celsius. Below 10 degrees Celsius, those are our psychrophiles. They can even flourish at what temperature is this? 0 degrees Celsius, which is freezing. These guys can live in your freezer. They can multiply and, in fact, favor growing in these freezing temperatures. And we're learning more and more about organisms that can live at freezing temperatures and, in fact, prefer freezing temperatures. Next, we have what we call psychrotolerant. These guys can tolerate freezing temperatures, but they don't prefer freezing. But they like it cold, but not too cold, okay? Psychrotolerant. Next, we have this green dotted line here, which are the mesophiles. These guys like middle range temperatures. We are mesophiles. We prefer it to be anywhere from room temperature up to maybe 45 degrees Celsius. We don't want it much hotter than that. And so anything that lives in that range or can survive like at fever temperatures, anything that lives at temperatures that you could survive at are mesophiles. Thermophiles like it hotter. So they're in our pink. They are here. These are our thermophiles. Now we're getting much, much hotter until you get to the extreme thermophiles. Remember, 100 degrees Celsius is boiling. And so we have microbes that will actually flourish at temperatures above boiling. That's wild. So we're going to learn a little bit about these groups here. We basically talked about the psychrophiles. They prefer the cold. They flourish in the cold. Psychrotolerant can grow slowly in the cold, but they don't necessarily prefer it below freezing. They still like lower temperatures. Mesophiles prefer the mid-range temperatures. Thermoduric are organisms that can survive short exposure to higher temperatures, things like pasteurization and stuff like that. So that's kind of a little bit of a higher range of our mesophiles, but they still prefer those middle temperatures just like we do. Thermophiles prefer it hotter, generally above 45 degrees Celsius, but usually still below boiling. But if you get up into favoring anything boiling or above, now we're talking about our extreme thermophiles and living in things like the deep sea vents and stuff like that. Some of our microorganisms will survive in certain conditions based on what's in their gas environment around them. This can rely on things like oxygen, nitrogen, and carbon dioxide. We're going to focus on oxygen and carbon dioxide, and specifically more time on oxygen. There are organisms that need certain percentages of carbon dioxide. If they do depend on that, then we can accommodate that for their living situations. But more importantly, organisms that fall into these categories based on oxygen exposure. We have organisms that use oxygen and therefore must be able to detoxify. Okay, if you use oxygen, you live in oxygen, you must be able to detoxify. And we'll talk about what I mean by that in just a moment, because you're probably like, what do you mean by detoxify? The process of using oxygen is nowhere near perfect. So there's a little bit of having to deal with byproducts of that that we have to be prepared for in our cells. So dependence on oxygen comes with having to deal with free radicals and things like that that can cause damage to your cells. So you have to have a protective mechanism in place to deal with byproducts of dealing with oxygen. We have organisms that neither use oxygen nor detoxify it. And then we have organisms that do not use oxygen, but can still detoxify it. So you must fall into one of these three categories. You don't use oxygen, you don't need to detoxify it. You don't use oxygen, but you can detoxify it. Or you use oxygen, you must be able to detoxify it. Okay. All right. So what do we mean by detoxifying it? We're basically talking about these byproducts of basically breaking down O2. And so we know, and we'll get reminded about this in chapter 10 when we review aerobic respiration, but in the electron transport chain, we are basically using water and H pluses and electrons. Water with electrons and half of an O2. So if we take 2 H pluses and 2 electrons and half of an O2, we can make H2O. And that's how we deal with our leftover H pluses and our electrons from the electron transport chain when we are aerobic organisms. Remember, this is happening in the electron transport chain in aerobic organisms. If you don't remember this yet, that's okay, because we're going to talk about it in chapter 10. We're going to review all that stuff, okay? Giving a little bit of reference on why we're even having toxic oxygen products. We can make them because this process of getting half an oxygen isn't always perfect. Also, in our environment, that can happen naturally. And so we have to be able to deal with those toxic byproducts of oxygen. So this leads to the presence of singlet oxygen, which is just an O, or it's also abbreviated as 1O2. This means that there's an oxygen that's got all the electrons that it needs and is happily living its life as just a plain old oxygen, but it's not super happy because it still can react with other things, including molecules in your own body. So this is an extremely reactive molecule that's produced by a lot of different processes in the environment, including produced by your own phagocytes as a defensive mechanism to kill invading bacteria. So we can actually use it to our advantage. But we have to be able to protect ourselves from it because it's in our oxygen-rich environment all around us. So if you are around oxygen, you might see singlet oxygen. You also might see superoxide. Superoxide is O2 that has an extra electron, or you might see hydrogen peroxide. So instead of just H2O, if you have another oxygen on there, then you could be H2O2. This is also reactive. And we also have hydroxyl radicals, OH negatives. All of these are other oxygen products that can cause damage to our cells, in addition to the singlet oxygen and potentially ozone and things like that. So all of these things can be reactive, and presence of oxygen type species in oxygen-rich environments are things that our cells have to be equipped to deal with. Some examples of how our cells deal with this. Superoxide dismutase. This basically takes superoxide, which is O2 negative, adds some H pluses, some protons, and creates hydrogen peroxide. And you might be thinking, we just said hydrogen peroxide is dangerous. Yeah, that's why our cells don't stop there. Then step 2 is taking the catalase enzyme, which converts hydrogen peroxide into water and oxygen. Anytime you have ever seen bubbles from using hydrogen peroxide, the catalase enzyme was breaking down the hydrogen peroxide into water and oxygen. Your cells can do that. So even if you see bubbling with hydrogen peroxide, your own cells could be doing that, not just bacteria. Some bacteria can do it and some can't, because we already talked about previously in this other slide, some microbes cannot detoxify oxygen. If they can't, they won't bubble from hydrogen peroxide, from that catalase enzyme. So there are some terms that we use to group these guys as well. Aerobes, or aerobic organism. An aerobe or aerobic organism, air, they use oxygen. Aerobes use oxygen, air, okay? So they must be able to detoxify. Remember, that was the rule. If you use oxygen, you must be able to detoxify it. So obligate aerobes are organisms that cannot live without oxygen, like you. You're an obligate aerobe. You will eventually die if we don't give you oxygen. So there are plenty of microbes, including bacteria, that are obligate aerobes. And we'll even see some of them in our lab as we're investigating our organisms in the second part of the lab after break and stuff like that. So anyways, it depends on what semester you're in when you're watching this. But yes, in our second part of our lab, the second 8 weeks or so, we will be looking at our unknown organisms. And some of you guys will have aerobes, but some of you guys might have anaerobes. And so what are anaerobe? Anaerobe means, an means without. Aerobe means oxygen, of course, oxygen dependent. So without being oxygen dependent. Facultative anaerobes are able to facilitate both oxygen and not oxygen. They can switch between. When oxygen is present, they use it, but they don't have to have it. They do not require oxygen, but they can use oxygen. When oxygen is present, they go through aerobic respiration, just like the aerobes do. When oxygen is absent, they go through fermentation. We'll talk more about what fermentation is in chapter 10, where they can switch between aerobic respiration using oxygen and fermentation, which is an anaerobic process that does not use oxygen. Okay? Then we have microaerophiles, little bit of air. They need less oxygen. Then we have anaerobes. Anaerobes are organisms that lack systems for using oxygen. So this can kind of get split up into that group of don't use oxygen, can't detoxify it, or don't use oxygen, can detoxify it. Okay. All right. Strict or obligate anaerobes. Anytime you hear obligate, it means like obligated. You have to do it. Okay. That's what obligate means. Obligated means you have to do it. So obligate anaerobes means they must be anaerobic. True obligate anaerobes cannot detoxify oxygen products. They cannot tolerate oxygen at all. So these are the guys that don't use oxygen and can't detoxify it. But what about the guys that don't use oxygen and can detoxify it? Those are called aerotolerant anaerobes. They're aerotolerant. They can tolerate air. They can detoxify it to a limited degree. So they can be present in the air, even though they aren't going to use the air. So they're anaerobes without air, but they're aerotolerant. So they don't use catalase. Typically, they use different processes to break down those reactive oxygen species. All right, moving on. So those are our gas requirements. We also have a dependency on like having certain amount of salt concentrations outside of the cell. Osmophiles are organisms that can have high solutes outside of them. And if they can have high salt specifically, then that's a type of osmophile called a halophile. Obligate halophiles must be in salty environments. So halophile, remember, phile means love, like philo, like Philadelphia. Okay. That means love. So halophiles love halogens, like chloride, okay, like sodium chloride. So halogens, those are her halophiles. So they like salt. Obligate halophiles must have salt. In fact, they have adapted to where they need the salt. Like they have adapted to their isotonic means more salt, and they can't survive in conditions that we survive in. So facultative halophiles can kind of tolerate have a high resistance to salt, but they don't normally favor high salt environments, but they can survive it if they need to. All right, we see a lot of microbes that are like this that might live on the skin, which is high salt concentration. They might be facultative halophiles. They can tolerate that higher salt content than what most other microbes can. All right, moving on. Organisms can have relationships with one another. Anytime that organism depends on that relationship for living, then that's symbiosis. So symbiosis is relying for survival on the relationship. But if you have a relationship with an organism that can affect one another, but it isn't required for survival, like you and your pet dog. Your dog can live out in the wild if it needed to. It's not going to die because you're not there. I mean, I don't know how your dog is doing, but my dogs were strays before I adopted them. So they were living out in the wild. And now I can feed my dog. That's a non-symbiotic relationship. It's what I would call a synergism. I'm benefiting by my dog being around, and my dog is benefiting because I'm feeding him and petting him and loving on him. That's a synergistic relationship. If it was a relationship where somebody's being harmed, but like let's say, I eat chickens. Okay, so that's an antagonistic relationship. I don't have to eat chickens. I'm not going to die without chickens. And chickens ain't going to die without me. But it's not going to benefit the chicken when I eat it, right? So that's an antagonistic, but those are non-symbiotic relationships. Once we get into a relationship that is required for survival, now we're in symbiosis, okay? In mutualistic relationships, both members benefit. And then in commensal, one benefits, but the other one's not harmed. Many of our gut bacteria fall into either mutual or commensal. A lot of the times our gut bacteria are benefiting from the food that we're eating, and many of them will break it down into products we can use. That's mutualistic. Or providing us with protection from our potential invaders by taking up seats now the invaders can't sit down in, taking up space, or producing components that would prevent other microbes from growing and things like that. It's called microbial antagonism, and we'll learn more about it in chapter 13. But if they're just living in us and benefiting from our nutrients, but they're not giving us back anything beneficial, that would be commensal. One of the most important relationships that we learn about this class is parasitism. When one organism is benefiting and then harming their host, that's parasitism. So most of the time, anytime you're infected with a pathogen, the pathogen is the parasite and you are the host. Parasitism is referring to this symbiotic relationship. Parasitism is a type of symbiotic relationship. For example, if we were talking about a viral infection like COVID, COVID has to live in your cells and use your cell's machinery and your cell's resources in order to multiply. And that's causing harm to you. COVID is a parasite and you are a host. It's a type of symbiotic relationship. All right, so those are the different types of associations between the organisms. These next slides just kind of break it down into a little bit further between our symbiotic relationships. Again, symbiosis is a relationship where their partnership is required for at least one of the organism's survival. It's not an optional thing. Mutualism, both organisms benefit. Many of our microbes that are part of our biome fall into this category. In commensalism, one benefits, but the other one isn't harmed or benefits. It just kind of exists. So the commensal organism is receiving the benefit, and the cohabitant is just living along with it. It's cohabitating. It's not affected. And then parasitism, one organism is benefiting while it harms the other one. The one that benefits is the parasite. The one that's harmed is the host. All right, another way that organisms, especially our microbes, can live together is through biofilms. Biofilms doesn't just have to be bacteria, but primarily usually associated with bacteria. It could be bacteria, it could be fungi, or it could be protozoa. Any of those can be involved in the biofilms. But typically what happens is our pioneer organism sticks to a surface, and then its secretions help other organisms stick on, and they secrete more things that help more organisms stick on. And then they can even communicate with each other about the health of their environment, especially fully formed biofilms as biofilms are generated. This allows for the organisms to basically sense what's going on with the biofilm and communicate with one another and say, hey, smells like food, let's keep going. Or, hey, smells like death, maybe we don't stay in this biofilm. So the ability of organisms to pick up on these chemicals, and they can even change their gene expression in response to it, hey, I smell food, we're going to start making these genes for breaking down food. Or, hey, I smell dying, we're going to start detaching and making less of our, fimbriae or whatever for attachment. And so all that can be governed in that quorum sensing, the ability to kind of communicate by chemical signals within the biofilm. So microbes can do that as a community sort of in a biofilm. So this is a pictorial representation of that. We have our pioneer organisms attaching to the surface. They create secretions that allow other microbes to join. They continue growing in size and sending signals saying everything's going good. And then once we start getting to a certain size, some organisms might leave because organisms are running out of food or whatever. That can lead to them becoming what we call planktonic or living freely, again, instead of being associated with the biofilm. One of the most common biofilms we can think of in biology and organisms like humans would be plaque on your teeth. That's a biofilm. So there's quorum sensing going on with the microbes that are on the plaque on your teeth that can allow them to communicate and say, hey, this is great. We're getting sugars and proteins and things like that from this person. And we're making acid that's breaking down their enamel, but it's just creating more surface for us to stick to. Meanwhile, causing you harm from that biofilm situation, which is why brushing and flossing is so important. All right, when our bacteria are multiplying, we learned in chapter 5 that eukaryotes divide through mitosis and reproduce through meiosis, right? Making gametes, and the gametes come together and that sort of stuff. But bacteria grow by binary fission. It is similar to mitosis, but we don't have a nucleus and we don't need like spindle fibers and all of that complicated stuff like we did in PMAT in mitosis. In bacteria, it's quite simple. They just make a copy of their genome, they expand the cell, and then they create a sort of septum, a band in the middle of protein that allows them to pinch off a new cell. This whole process is called binary fission. One cell becomes two equal daughter cells. And so basically the division of cells through binary fission in bacteria, one cell becomes two, two becomes four, 4 becomes 8, 8 becomes 16, 16 becomes 32, and so on and so forth, just like with PCR, right? Instead of DNA doubling, now it's cells doubling. And so this process, how long it takes an organism to go from 1 to 2 cells, determines how quickly it's going to take for your culture to grow up too. Some organisms double very slowly. So that doubling time, going from this number of cells and then doubling that number of cells, that doubling time can be a predictive thing for identifying the organism, but can also be constraining in studying them. So many mycobacterium, remember like Mycobacterium tuberculosis or Mycobacterium leprae that causes leprosy, and other Mycobacterium organisms grow very slowly. We're talking a doubling time of a month to 2 months. So going from one cell to just two cells in like 2 months. That is wild. And so culturing microbes like that is almost nearly impossible. So you almost have to study them in disease states in like animal models and things like that. Very difficult to culture those guys and study them appropriately. So we've had to learn to work with them in different ways. But then you have other organisms like E. coli that have a doubling time of like 20 minutes. So it just depends on the microbe. And many of them are more like on the E. coli end. Many of them have a doubling time of like 20 minutes to an hour. That's the most common of what we see with our typical pathogens, our bacterial pathogens. And so in order to start doubling, though, remember that the real process that's going on when we're doubling our cells is that first you have to replicate your DNA. That takes some minute, okay? It's not like instantaneous. And so if you have a collection of cells, you've taken your sample from your cloudy liquid and you've mixed it into fresh, sterile liquid. The first thing that has to happen, the cells have to realize they are in a situation where, oh, we can start multiplying again. They have to make copies of their DNA and then, split up off the cells and everything like that. The time that it takes to go from being introduced to the new environment and then actually starting to double your cells is the lag phase. You don't immediately start doubling the cells. It takes some time to get to that doubling. Again, if that bacteria takes 20 minutes to go from one cell to two, that 20 minutes is our lag phase. Okay, so it's the amount of time it takes for that cell to start making copies of itself and realizing that it can make copies of itself. So that's the lag phase or the lag period in our bacterial growth curve. All right. Once we start doubling and keep doubling and keep doubling and keep doubling, that's the exponential growth phase. We got plenty of nutrients. We can just keep doubling and doubling and doubling. Once our growth and our ability to keep growing starts equaling cells just dying because they're old, we get to the stationary phase. It's like our maintenance phase. We've maxed out on what that environment can tolerate, and growth equals death. But once death starts outweighing, we start running out of nutrients or we start making toxic products from dead cells being around, then that's the death phase. We'll see that the disease progression kind of mimics this. When we get into chapter 13, and then we get into talking about disease and infection, we'll see that the disease phase kind of mimics our bacterial growth curve, our microbial growth curve, okay? So again, that lag phase is the period when that population is kind of adjusting to the new environment and starting to decide to make copies of the new cells and just beginning to make new DNA and things like that. All right. Then our exponential phase or our log phase. Log means logarithmic. It's our doubling the previous round every time is exponential growth, also known as logarithmic growth, okay? So this is when our growth is increasing dramatically and we're increasing that population. We've got plenty of nutrients. The environment is favorable for continuing to just grow new cells. Once the cells need to stop growing or death and growth start equaling out, we're in survival mode. That's our stationary phase, our maintenance phase, if you will. And then our death phase is once the death rate starts to significantly outweigh our growth rate, and we start losing out on nutrients or we have waste products from dying cells building up. The death phase, the cells start dipping more and more downward, more drastically over time. All right, so that is it for chapter 9. And let me know if you have any questions. Happy studying.